Yes, so thank you so much for the very kind introduction. Um, so I'm gonna go ahead and kind of discuss the technologies we recently developed in my laboratory over the past couple of years to focus on uh, sort of the spatially resolved multi-omics mapping from sort of genome transcriptome on the way to proteins, and in particular how we can combine them, but combine them not for fun, but really to help understand the complex process in tissue development, aging, or different disease, including cancer. Um, so this is my disclaimer. And so how we kind of get started with this kind of single cell or spatial, uh, really back to 2006 when I switched my own research career from physical science to biomedical research. When I started as a postdoc back to 2006, I read a number of papers. There's one of those written by Professor Joanna Joyce, talking about the complex tumor microenvironment. And I was very fascinated by reading this paper. I realized, oh, if you really want to understand what the tumor microenvironment actually is, you need a technology that can resolve the single cells, number one. Number two, ideally, you can kind of understand what's going on directly in the tissue context rather than sort of completely dissociate the tissue into individual cells. But let's do that step by step, kind of the early days, even though you want to do single cell high-plex analysis of proteins, that's not, no technology exists to allow you to do that. And so, okay, can we develop that technology first? So single cell high-plex protein measurement analysis. So that was kind of first a couple of years when I even after I moved to Yale, and I work on the single cell immune function profiling technology. So I think a couple of amazing students and a postdoc in my library contribute to this work, including my very first graduate student, Min-Shu Kwan, and now he's assistant professor Yang, and my associate research scientist currently in my lab, Dongju Kim, who took over this project. And so later, after Min-Shu left, so in this study, we were able to map down kind of a dozen of to kind of 3 dozens of proteins secreted from single immune or tumor cells. We found actually, for example, in normal bone marrow cells, you see very tight clusters of those protein secreting cells when you sort of cluster them based on the protein secretion profile. But when you look at the leukemic bone marrow microenvironment, the cells secreting a lot of proteins, and they're not supposed to produce. And as a result, you see much more polyfunctional cells, so really spread out in all over the map. So we still work on that. I think we further expand this study to immune cells, for example, the engineered CAR T cells, and that has been our interest even until now, since even though we moved to spatial, pretty much in majority of the lab moved to spatial, we still work on this kind of single cell analysis. But as I mentioned in the very first slides, of you need a tool that can measure single cell omics. But ideally, that's in a spatial tissue context. So people realized that challenge about 10 years ago and started to develop a range of spatial omics mapping technologies. So broadly speaking, there are 2 different directions to develop the technology. One is that's called imaging-based or image-based. And so 10 years ago, if you can image like a 3 or 4 different fluorescent markers, and that's pretty cool. But nowadays, for example, SIGFISH and MERFISH can image up to entire transcriptome, like 10,000 genes, using combinatorial barcoding plus sort of cyclic multiple rounds of imaging. And another direction is instead of doing this repeated imaging over and over, can we use high throughput NGS sequencing to map down the spatial omics? In most cases, spatial transcriptome, but I would like to say it's not limited to transcriptome, definitely not limited to spatial gene expression. And so the history of this NGS-based spatial omics is relatively new, and start with this landmark paper published by Joachim Lamberg to use the DNA microarray to capture messenger RNAs released from the tissue section placed on top of the glass slide. But since then, this has been commercialized at 10x Visium, I think everyone knows. But many other groups further pushing the boundary to make the spot size much smaller, such that you can even do kind of subcellular spatial transcriptome sequencing. But they all follow the same fundamental principle Professor Joachim Lamberg coined called barcode solid surface for spatial transcriptome or for messenger RNA capture and spatial transcriptome sequencing. But what my laboratory came up with a couple of years ago was totally opposite. Okay, instead of capturing the messenger RNAs or whatever molecules to the barcode surface, we deliver those DNA barcodes into the tissue, and the tissue itself is the reaction chamber. We can perform all kinds of reactions there. And eventually you can look at gene expression, you can look at proteins, you can look at many other things. And so after we published this paper back to 2020, so many different research groups actually adopted technology, and they were able to even make the technology even better, higher data quality. And also you can tweak this cross-flow barcoding to make the DNA microarray. That's kind of like a custom DNA microarray. If you want to use the Visium workflow, yeah, you can still use the Visium workflow, but you use the DNA microarray you'll create it by yourself using this same type of microfluidic device we developed. And so how the technology works seems like a lot of people already know. I don't need to go into very tiny details of this entire workflow, but kind of at a high level, we use the microfluidics to deliver DNA barcodes into the tissue, but you can use other methods as well. And so what we did is, if you have the tissue on a glass slide, you can use the first set of microfluidic device to deliver DNA barcodes A1. Now we do A1 to A100, and those barcodes can detect the messenger RNA, so can detect the proteins or other things, and then you use another microfluidic device to deliver the second set of DNA barcodes. And so after you ligate them together, you create this 2D grade of tissue pixels. Each has unique combination of A and a B, so that's how you get sort of spatial resolve address code for individual pixels, but you can map down all those pixels across the entire tissue section, spot by spot or pixel by pixel. And once you use the parent sequencing, one end you write down the spatial address code, on the other end you write down what that gene is. So that's basically how the technology works at a very high level. But recently we really try to make that work so well for clinical tissue specimens. I think Gadi gave an amazing talk, and also how to analyze the different mutations, tumor clones. And we keep that in our mind. We believe that's so important in a clinical. So we ask ourselves, can we make our technology fully compatible with FFP tissue samples? And I show you a couple of examples that works. But what I want to say is not only does it sort of worked out very well, but the data quality actually could be even better using this in-tissue barcoding approach. For example, this mouse brain, we were able to detect over like 30,000 UMIs per pixel, and almost 7,000 genes per pixel per spot. And that's the spot size 50 micron, which is equivalent or actually slightly smaller than 10x Visium. But we're comparing the data to 10x Visium based on a probe base, not a base by base sequencing. We can get a comparable data quality or even slightly better. And so we're also looking at this human patient, a lymphoma patient, FAP tissue specimen. We got extremely high quality data on the clusters associated with different kind of tissue types, including malignant and non-malignant. And as I mentioned a little bit, so can we, since we're doing base by base sequencing, can we really detect those mutations? I totally didn't anticipate it gonna work, but it turns out it worked so well. So we're able to detect on a kind of somatic mutations and also germline mutations. And so from the same tissue, you can quantify the total number of mutations you detected. That's sort of like a mutational burden, right? Tumor mutational burden. So in this case, we can use that to see where you see higher mutational frequency, where it's lower, and a higher mutational frequency indeed corresponds to the tumor region. So now based on this de novo mutational calling, you can clearly identify the tumor region. You can also use the mutational profile to cluster on the different cell types, on the different tissue regions showing here. So we know those are tumor, those are more like at least most of the cells in here are kind of non-malignant, and then we can kind of separate them to look at all the mutations across the entire genome, across all 22 chromosomes. And so this basically gives you this tumor versus non-malignant versus malignant, but you can also further sort of divide the malignant tumor region into different subclones based on the mutational landscape. I'm not gonna go into the details, but this is something I was so excited about recently, so I just cannot stop kind of sharing this exciting latest development in my laboratory. So when we published that paper back to 2020, so the Nature Methods highlight our work and saying, hey, the multi-omics sequencing goes spatial. So now the question is, that time we were able to detect about like a third proteins in conjunction with the whole transcriptome, but honestly, that still doesn't meet my standard. If you want to call proteome, at least I would say you should be able to map down hundreds of proteins. And then we say, okay, can we do that? I have no idea, but just give it a try. It turns out that does work and it worked out quite well. For a variety of mouse tissue types, we were able to map down more than 200 proteins in conjunction with whole transcriptome. And for human, we were able to get more antibodies to create this panel. So now that's even over 300 proteins in conjunction with entire transcriptome. So if you're interested, you can check out the paper where it's published, but we're making the panel even bigger and bigger nowadays. What is unique in this kind of spatial, we call spatial seq, is compared to single cell seq or single cell transcriptome sequencing, you don't detect anything outside cells because you dissociate the cells, you get either single nuclei or single cells. Anything outside the cell, like extracellular components, they're often called in the dark matter of your single cell sequencing data. But in our spatial seq, we can also map down those extracellular matrix components, extracellular matrix proteins. And for example, here we combine 16 extracellular matrix proteins in our panel to do this ECM inclusive spatial seq. So you can see all different cell types, tissue types, and based on transcriptome, based on the protein profile. But now you can also see where the different ECM molecules are located, and you can really see how the ECM molecules interact with different cell types. And so what we have been very interesting from very beginning when I started to to think about this technology, to develop that technology, I was telling myself and my group very clearly, we're not inventing another spatial gene expression or spatial transcriptome technology. Instead, I believe our in-tissue barcoding approach is very versatile. You can do many other things. In particular, you can look into the epigenetic mechanism spatially, spot by spot across the entire tissue area. And we decided to look at the first two epigenetic mechanisms. One is the specific histones modification. The second is global chromatin accessibility. So for global chromatin accessibility analysis, very elegant chemistry developed by Howard Chang and Will Greenleaf at Stanford called ataxic. So what ataxic does is use the enzyme called TM5 transposase to process your tissue. But in most of the cases, okay, nowadays, if you do single cell ataxic or do even bulk tissue ataxic, people always tell you the first step is to dissociate the tissue, get a high quality nuclei, kind of purify and collect high quality nuclei. But we found totally unnecessary. Okay, if you have very thin tissue section you place on a glass slide, you can just go ahead and do TM5 transposase to train the tissue. And what this enzyme does is gonna cleave the genomic DNA and open chromatin region, and also you pre-assemble the DNA oligo into this enzyme, this TM5 transposase. And once TM5 transposase cleaves the open chromatin region in the genomic DNA, it's gonna insert that DNA oligo into this open chromatin region DNA sequence. So eventually, if you collect it on a genomic DNA and amplify and the sequence, you look for those DNA oligos inserted into the DNA, genomic DNA. You can map down where across the whole genome are kind of more accessible, less accessible. So that's how the ataxic works. But in our case, after we performed this TM5 transposition, we did two more steps to ligate the spatial barcodes A and the spatial barcodes B, and then we can basically one experiment. We do thousands of ataxic, and by each experiment you do basically spot by spot or pixel by pixel, and so you can collect all the genome-wide ataxic data traces from all those individual pixels. So we can aggregate some pixels, for example, here associated with embryonic mouse liver to produce this pseudo-bulk ataxic data trace. So now you can see this gives you a very strong concordance when you compare and benchmark against the encoded reference data. And in our case, somehow we see even slightly better signal to noise ratio, see those very, very sharp peaks. And so once we have all the pixels on the spots for ataxic genome-wide ataxic analysis, you can use the ataxic profile to cluster on the pixels, very much like how you do a single cell ataxic. But what's different here is I think every pixel, every spot here, after you do the clustering, you see all different cluster, but you know exactly where they are, right? So you can go back to see those pixels where are they in the tissue. And for example, this cluster, very tight cluster, is they are in the liver, and this cluster extremely tight, and they are associated with the spine. And so you can do your own kind of annotation. You say, oh, that's indeed hepatocytes or some kind of urethral progenitor cells or red blood cells that are in the liver. And but you can also go back to see exactly where they are. You can look at those individual genes. For example, this guy, okay, this gene SPTB is a erythrocyte or urethral progenitor cell gene, but in the embryonic mouse, in the mouse embryo, actually liver is no organ to produce those red blood cells. So now you see exactly this gene is much more accessible in a liver region. And so you can look at many other genes as well. In particular, in brain, you see, oh, many tissue types already started to develop, and the different neuronal cell lineages, the progenitor or towards the accessory neurons and a more mature accessory neurons or some kind of neuronal progenitor cells. You already see all those different kind of brain tissue types started to develop at this stage. That does look like a gene expression, but just want to let you know this is not gene expression, right? You can directly visualize the spatial sort of the accessibility of the chromatin state, okay, in across the whole tissue section. So again, we benchmark against the ENCODE reference data across all different major organs. You do see pretty good concordance, and our data gives you much more granular. Because we mapped out spot by spot rather than the whole organ, we cut it out and do a taxic, right? So that makes sense. And so what is much more powerful is once you have the data from single cell RNA sequencing or single cell ATAC, and you do on cell type annotation, it's a lot of work, but you can get that done, or people maybe have done that before. So now you can integrate your spatial ataxic data with the single cell RNA sequencing data by doing this co-embedding. And then you can look for example, those black dots, those are the data points from our spatial ataxic experiment. And so if you see those black dots overlap with those kind of, for example, those blue dots, and you know, okay, the dominant cell type in those pixels must be this particular cell type, then you go to look for what that type of cell is. So this is kind of pretty straightforward to understand how we do this cell type annotation using this kind of co-embedding and a label transfer. So you can pick a couple of those and visualize where the different cell types are in the tissue. I just show you a couple of those, like for example, urethral lineage, the definitive urethral lineage, red blood cells, you see they're strongly enriched in liver because liver is an organ to produce red blood cells at this stage. You see like accessory neurons and some pre-mitotic, premature neurons, and wow, very distinct neuronal cell types. And now you can see where they are. But keep in mind, what you are seeing is not just where they are defined by the gene expression profile, but actually where they are defined by their epigenetic state. That might even precede the developmental stage for that particular cell type if you define a cell type based on transcriptional. So that gives you a different message, but very, very important, and that might even give you some dynamics, developmental dynamics message. So recently in my laboratory, we are very interested in the the other side of the whole story. Okay, not the developmental process, but the aging process. In particular, how senescent cells play a role in aging and potentially in disease as well. So from our data, since this is unbiased genome-wide, you can look for those senescence-related marker genes, for example, or those kind of P16, P21, and other markers associated with different biological pathways. Here we look at a human liver, and from our spatial ataxic data, you can clearly see the different zones, kind of zone 1, 2, 3, kind of from the portal vein to the central vein, and but on the pixels, you can look at those senescence marker expression, right? Like gamma H2AX associated with some DNA damage repair mechanism, and this two are P21, P16. So now you do see very distinct spatial expression pattern for, not spatial expression, sorry, the spatial accessibility pattern of those senescent marker genes. And really highlighting the heterogeneity of the senescent cells or senescent-like cells that may not be senescent, but they are kind of upregulating those pathways, potentially can drive them to become senescence. And when you look at epigenetic, you may capture that transition even early on. so once you can do global chromatin accessibility analysis, and we ask, can we also look at the specific histone modifications? It turns out it's totally durable and the same chemistry using TM5 transposes, but a little bit tweak before you apply TM5 transposes, you use an antibody to recognize the specific histone marks first, and then using the protein A tether TM5 transposes to process to treat the tissue, and then where the cleavage site's gonna be in this case should be associated with the different histone modifications rather than just a global open chromatin region. So now after your sequence, the results basically are associated with the specific histone marks. And so now we did this for 3 different histone marks. You can see different histone marks sort of mediates the development of different tissue types, in different regions, but different histone marks have slightly different role during the developmental stages. For example, this histone mark really can now differentiate the tissue types here and there, and I would not say that histone mark is not capable of differentiating different cell types. That's not a valid question. The answer should be, okay, this histone mark that's not really mechanistically, that's not really control the development of those distinct tissue types between here and there, but other histone marks gonna do, right? So now what you see is the differential role of different histone modifications in the cell tissue development process, or what type of tissue gonna develop at this stage is controlled by what type of histone modification. So that's the message and again, you really learn the biology rather than just the cell type or tissue types. So now you can visualize individual pixels perform unsupervised clustering as what I described before, but after clustering, you can see where there are just very kind of nice clusters corresponding to the tissue regions, and so you can visualize some specific genes as well. But keep in mind, you are not just seeing gene expression, but the message should be what you see is whether or not the K4 trimethylation actually controls the activity of that particular gene in the certain tissue and in certain specific tissue region. So that's the complete message you get. And so not just a cell type, tissue type, but the mechanism, whether or not that histone modification does control or maintain the tissue development in that particular region. So again, if you do integration co-embedding with single cell reference data, either single cell RNA-seq or single cell epigenome sequencing, you can transfer the cell types from the single cell data annotation, and you see where the different cell types are. But again, I want to emphasize is what you see is not just different cell types in the tissue, but actually whether or not the histone modification K4 trimethyl plays a role in the development of this particular cell type at this developmental stage. All right, so once you are able to do spatial epigenome and spatial transcriptome, and the question, can you really do both, which is not just for fun, it's very important because hypothetically you see, oh, the epigenetic regulation really kind of controls the expression of the genes, but can really measure what genes are expressed and then correlate with the underlying epigenetic mechanism, right? And so we demonstrate that works so well, and we demonstrate that for both spatial ATAC plus whole transcriptome, but also spatial histone modification, spatial cut tag, and whole transcriptome sequencing. And so I just want to use one example to show. And so when you have both, what's the added value? So for example, this developing mouse embryo, and so when you use this epigenome ATAC data from your co-mapping experiment, or use the RNA data from your co-mapping experiment, you can always perform the unsupervised clustering to identify different tissue types. Seems like either one works well, and the transcriptome can resolve a little bit more clusters, but either one can define the major tissue types. And again, you can also do all this kind of co-embedding to transfer the different cell types and visualize where they are, and they're very similar to what I described in a spatial ATAC or spatial kinetics data analysis. But what is interesting here is, so now once you have both, you can really examine some interesting biological process. For example, the differentiation of neuronal, sort of the progenitor cells, in this case, radioglia, to post-mitotic premature neurons. And you can further look into the differentiation towards the etc. neurons as well, but we decided to just look at this specific step from radioglia to post-mitotic premature neurons. And you can look into this differentiation process in a shorter time based on either RNA or based on ATAC. And the simple hypothesis is when you see the chromatin opened up, and later you're going to see that gene being expressed, right? So because for gene expression, you need to open up the chromatin first. And that seems to be true for many genes, but not always. And so, for example, this particular set of genes, the chromatin opened up, and chromatin remains open for those genes. But those genes expressed at the very beginning, but quickly diminish. They're not expressed anymore. And then when you look at what are those genes, turns out they are very interesting genes. They are not associated with the neurons. They're associated with oligodendrocyte or oligodendrocyte progenitor cells. I think now the take-home message from this analysis is at least some of those cells, once they're committed to neuronal lineage, they're going to become neurons. But they still maintain the epigenetic capability or epigenetic potential to become kind of oligodendrocyte lineages. So that's something very interesting. Only if you have this kind of multimodality, multi-omic data, you can tell that story. So we further kind of analyze those sort of differentiated cells and see where they are, and based on either transcriptome or epigenome. Now, based on epigenome, you see indeed that there's a bifurcation. So there are two branches. So one corresponds to some sort of cells that they're committed to neuronal lineage, but they still maintain very high level of potential to transdifferentiate into oligodendrocyte potential, but the other is more towards the neuronal lineage. You see the very clear bifurcation over there. So recently we're able to further combine proteins to do this spatial tri-omic mapping. So epigenome, transcriptome, but also hundreds of proteins. And what is more interesting is many of those proteins are associated with immune response, like immune cells. And so now we are applying this spatial triomics to the study of kind of neuroinflammation and neurological neuroinflammatory diseases or some autoimmune disease in brain. And so I'm going to skip those, just give a final kind of perspective, what the future should look like, at least in my laboratory. We're very proud of those spatial multi-omic sequencing technologies we develop, but we also believe, okay, you need to kind of integrate with the spatial imaging data to really kind of get the best out of both. And when you combine that will be much more powerful than single modality data. So we're doing that by combining spatial transcriptome and the sort of high-plex immunofluorescence imaging such as Codex, but we also further combine that with the spatial analysis of the metabolic activity that tells you the function of individual cells, right? So this is one of the tissue region we mapped out using spatial transcriptomics, but in the same tissue we also have the high-plex immunofluorescence using about like 50 different markers. I just show you 3 of those, but actually have data from 50 different markers. So now you can see all individual cells. There's a B cell follicle. You can see there's a light zone, there's a dark zone, but from the same tissue, we have the spatial metabolic analysis, or what I believe is there's a redox ratio telling you there's a glycolysis versus oxidative phosphorylation. So people already know kind of glycolysis activity is high in a light zone. And so you do see indeed, okay, in a light zone, you have the higher redox activity, and always light zone, and a light zone here. That people know that, but people never know, okay, what cells actually operate at those higher level of redox activity. Now we can see, and they're now very uniform. This is pretty heterogeneous. All right, so I'm gonna just stop here. I hope still some time to answer your questions. I don't really need to summarize. We develop those technologies, but I believe we develop those technologies for purpose, not for fun. And the question is, where are interested in my laboratories? So one is really kind of immunology in the context of sort of aging, and also cancer and senescence. And the second area in my lab is the brain neuroscience and sort of the cells, the taxonomy and the cell map in the brain tissues. And so I would like to thank the people in my laboratory and the funding support. And thank you so much for your attention. Even virtually, I can visit this beautiful campus. I really appreciate the opportunity to speak. Thank you very much.
Okay, thank you very much. So there are some questions. You can upload your question in the website or a chat window, or some can ask by in the Zoom with your mic. One question uploaded is already, you can see in the Zoom in the slide, the immunizing, on your immunizing, you mentioned changes in attack peaks with senescence. Were these changes in the same type of cells? Did the changes in chromatin accessibility vary from peak region to peak region, or was there any overall trend of changes seen? You can read it, right?
Yeah, that's I totally agree. I think that's why we want to kind of combine the multiple modalities to really pinpoint exactly what the cells contribute to the senescence and contribute to, those all metabolic activity, right? And so we are getting there, but that data is still pretty early. I don't want to kind of make a very concrete conclusion right now, but I would say majority of those are hepatocytes. So very likely what we're seeing is really kind of senescent hepatocytes, but in different zones, right? So the liver zonation, we notice the hepatocytes in 3 different zones. Actually, they have much very distinct sort of tendency to become senescent once they're under stress. And so I think it's still too early to tell you the full story. But this is really sort of the capability the technical capability I want to share with you.
Okay, the next question is the segmentation. I think the most important thing about the spatial transcriptomics is how to segment the cells. How do you manage to segment the cells into single cell?
I would not say we can sort of do cell segmentation using our spatial transcriptome. This is very much like a 10x Visium, although our spot size is much smaller, but still not a subcellular. Even subcellular, I still don't think that's the perfect data to do cell segmentation to get a kind of truly ground truth type single cell transcriptome sequencing. But there are many different computational methods out there to allow you to do cell type deconvolution or decomposition. And so we recently fun. Actually, you can do spatial transcriptome sequencing or spatial epigenome sequencing and the high-plex immunofluorescence such as Codex on the same tissue, the same cell, and afterwards you can see exactly what cells you have in every single spot, and that allows you to do a much better job to split that gene expression profiles of that transcriptome from that spot into different cells, because you know what cells, what type of cell you have. Now you just kind of split that transcriptome to those individual cells. Turns out that worked out so well. I didn't share any results from that analysis, but so that worked out, and I believe that's really the best way to get a kind of truly single cell spatial omics, either transcriptome or epigenome.
And the next question, she asked many questions in the paragraph. So how many antibodies have you tried? Is there any error rate, the confirmation of quality, and so any limitations? So can you ask the question? Can you answer the questions?
Yeah. I think how many we have tried, that's pretty straightforward to answer. About 300, 305 or 308 each. But I want to say that's not a limit. Instead we use only like a 10-20% of the sequencing length in a Illumina flow cell to sequence those, to map out those proteins. So that means in theory, you can really map thousands of proteins. That's still durable. But I do agree, when you co-stain thousands of antibodies, you may have some challenge, so that they're competing for the space in the cell, right? You may have much lower sensitivity to detect lower bounded proteins. That's always the case. On the other hand, and how to optimize those antibodies in a panel to make sure every antibody works, I was in no way to make sure for academic laboratories. But good news is I think industry industry makes the commitment that they develop those cocktail, they validate in different tissue types. They already did all the titration, carefully titrate the antibody to optimize the concentration. We kind of piggyback what they have done and apply that cocktail to our spatial. But on top of that, we added the antibodies on the proteins were interesting into our panel, and so that's kind of our strategy to do this high plex. But I would say this has huge potential, and the industry should have taken the lead to develop this. It's a totally durable in antibody companies, but very challenging for academic laboratories.
Okay, and 3 more questions uploaded. The next question is about co-filing of RNA proteins. So the intra-signaling protein or ECM protein in spatial omics, is there any difference?
Yeah, I understand the question. I think intracellular proteins, signaling proteins, they are much more difficult to detect now, oftentimes because the antibodies are not that high affinity, and in particular for those phosphoproteins. But recently, I think my collaborator Rahul said here at the New York Genome Center that they developed the phosphocysic, and that worked out. So now we're collaborating to do spatial kind of phosphoprotein plus the whole transcriptome, so phosphospatial-cysic, yeah.
And next question, how many histone modification plexes do you think is possible for spatial contact at the same time?
Oh, yeah. I didn't make it clear. Actually, right now, this is single plex. We didn't multiplex, and turns out this is now that straightforward to multiplex. But my collaborator, Gonzalo Castello Branco, they published a paper a couple of months ago to do 2 using antibodies for single cells. I believe that should work for spatial contact. We just haven't tried using those nanobodies. But it's not that easy to truly multiplex. But as I said, even you don't multiplex, because this is now single cell, right? This is really spatial. You can take the several tissue sections and do each section you do different histomodification, but once you put data together, you can see to some degree this kind of multivalency or how different histomarks work differentially or maybe kind of together to control certain tissue type development. Yeah.
And she also asked the question if would it be possible to look at the DNA methylation along with the histone modification?
Yeah, it is possible. I think one of my former postdoc, now assistant professor at Penn, he's very interested in his work on and we brainstorm. We know that they should work, and I'm now doing this in my lab right now. But yeah, if that's the technique you are interested in, reach out to Yanxiang then at the University of Pennsylvania.
Okay, the next question is that among the modalities, attack and different histone modifications, what is the most spatially well-resolved modality?
What is the spatial?
What's the
spatial well resolved. Oh, I think in our study we use pretty much the same spot size. Yeah, basically the same spot size for all different modalities. But I guess maybe you were asking, like for epigenome, you are looking at a nucleus, right? So in that case, if the nucleus is in a pixel, and so that might give you a little bit higher spatial resolution or spatial distinction, because yeah, you don't care the sort of the region between nuclei. And I agree, and that's kind of what we saw in our data. Often the spatial attack or spatial spatial epigenome data gives you pretty distinct sort of the separation of different tissue regions, even like a single cell layer. That's a pretty clear. But transcriptome data always a little bit, kind of fuzzy boundary in between.
Because time limit, this is the last question. Have you ever tried integrating all three modalities, the attacks peak at RNA expression level and protein abundance exhibit different distributions? Is there any difference in normalization for each modality?
Oh yeah, I show you a little bit data, all three modalities combined. But even when we kind of mapped out RNA and the protein, we saw the correlation between protein and transcript is not consistent at all. That's real biology. That's not a problem. And but the one I show you, one of my slides, there's one particular gene, you'll see strong concordance, very consistent across all three different layers, but not always that case. Yeah. Yeah, that's what I can tell, and but I don't have very detailed quantitative statistics. How many of those give you very constant? It depends on how you pick those proteins as well. And oftentimes the surface markers are relatively more consistent across the different modalities, different layers of omics. But if you're looking into more functional, for example, the protein secretor, to do some specific immune effector function, that correlation is much lower.
Okay, thank you very much. Thank you for your wonderful talk and the questions, the answer the questions in very late time in your zone. Thank you very much.